Have you ever wondered why you're awake right now, like listening to this in the daylight rather than sleeping in a burrow somewhere waiting for the sun to go down?
I mean, it's something we totally take for granted, right?
Oh, absolutely. Well, welcome to our deep dive. We have a truly fascinating topic on the table today, which is this evolutionary and biological switch that turned early mammals from nocturnal creatures of the night into, well, diurnal, day-walking beings like us.
And to figure this out, we're looking at a really landmark 2026 research article published in the journal Science.
Yeah, by Beal and colleagues. It's titled A Cellular Basis for the Mammalian Nocturnal-Diurnal Switch. And our mission over the next little while is to completely unpack every conceivable mechanistic detail of how your body's cells inherently know what time it is.
It is such a wild paper, too.
It really is. We are going to explore the surprising thermal and metabolic levers that control this biological switch and how the food you eat might actually be messing with your internal clock.
The mechanisms detailed in this paper are staggering because they touch on something so fundamental. You know, at a cellular level, your body is performing some incredibly complex thermodynamic acrobatics just to keep you functioning in the sunlight.
Right, and to really appreciate those acrobatics, I want you to picture this. It is the Cretaceous period. The sun is blazing, the air is thick, and the daytime world belongs entirely to the dinosaurs.
Yeah, it's not a safe place to be.
Not at all. If you are a tiny, early mammalian ancestor, you absolutely do not want to be out in that sunlight. I mean, it's a death sentence. So you hide.
You wait in the shadows for the sun to set.
Exactly. You wait for the temperature to drop, and only then do you scurry out into the cold, dark night to hunt and forage. You are strictly, fundamentally, a creature of the dark.
Nocturnality wasn't like a lifestyle choice for our ancestors. It was an absolute non-negotiable survival imperative. The daylight was spoken for by massive predators.
But then 66 million years ago, the asteroid hits.
Boom, the non-avian dinosaurs are wiped out.
And suddenly, those dangerous sunlit hours are completely empty. The evolutionary real estate of the daytime was vacant, and those scurrying little mammals rapidly expanded into the light.
Which is amazing, but it creates a massive scientific headache.
Okay, let's unpack this because there is a massive biological paradox right at the center of this transition, and it's the core of the science paper we're looking at. Mice, for example, are nocturnal. Humans are diurnal.
Right.
But biologically speaking, we share the exact same cell-autonomous circadian clock mechanism. Our internal timekeeping machinery is identical.
Down to the genes.
Yeah, so how on earth does one species love the night while the other thrives in the day if we are using the exact same biological clock?
Well, solving that paradox requires us to look at the anatomy of timekeeping. We really need to locate where this nocturnal-diurnal switch actually lives in the body.
Okay, so where is it?
Well, in all mammals, the master clock is a tiny localized region in the brain called the suprachiasmatic nucleus, or the SCN.
Suprachiasmatic nucleus.
Exactly. It sits right above the optic nerves. Its entire job is to take light cues directly from your eyes and tell the brain what time it is.
So the SCN is taking in the literal sunshine.
Precisely. And the fascinating part is that the SCN operates identically in both mice and humans.
Wait, really?
Oh.
Identically?
Yeah. Regardless of whether the animal is a night crawler or a daywalker, the SCN peaks in its electrical activity during the daylight hours. It always encodes daytime as active.
So the brain's clock is universally diurnal.
Yes.
That makes the paradox even wilder. I mean, if the brain's clock is the same, there has to be rebellion happening somewhere else in the body, right?
That's exactly what the researchers figured.
The peripheral cells, like your skin cells, your liver, your lungs, they must be interpreting that central signal completely differently.
Yes, there has to be a disconnect.
Let me try an analogy here. Imagine the SCN in the brain is a master conductor of an orchestra, and the peripheral cells in the body are the musicians.
Yeah, I like this.
The conductor is standing at the front, waving the baton, playing the exact same sheet music for both the mouse and the human. But while the mouse musicians have instructions to play loudly on the downbeat, the human musicians have biological instructions to play on the upbeat.
That is a perfect way to visualize it.
So they're reading the same sheet music, but their timing is completely, fundamentally opposite.
And this idea of an opposite timing mechanism is deeply supported by the data in the paper. The researchers suspected there had to be some kind of signal inverter between that conductor in the brain and the musicians in the body.
A signal inverter. Okay, how did they find it?
They designed a brilliant in vitro experiment. They took primary fibroblasts, which are basically just basic skin structural cells from both humans and mice, and they grew them in petri dishes.
Okay, skin cells in a dish.
Then They subjected these isolated cells to strict 12-hour temperature cycles.
Wait, why temperature? I thought the master clock runs on light.
It does. But deep inside your body, your organs can't see the light. What they do experience is that your core body temperature naturally rises and falls every day.
Oh, right. You warm up when you're awake and active, and you cool down when you sleep.
Exactly. The researchers simulated this by putting the cells in incubators, 12 hours at a warm 37 degrees Celsius and 12 hours at a cooler 32 degrees Celsius.
So they're changing the temperature up and down, but how do you actually see a biological clock ticking inside a microscopic cell? I mean, it's not like there's a tiny clock face on the membrane.
No, but researchers use something called bioluminescent reporters.
Okay, what is that?
They take the specific genes responsible for the circadian clock, genes with names like BMAL1 and PER2, and they attach a glowing bioluminescent tag to them.
Wait, so the genes actually glow?
Well, the proteins they make do. Every time the cell manufactures one of these clock proteins, it literally emits light. You can put the petri dish under a special camera, and you can watch the cells glow brighter and dimmer in a rhythmic cycle.
That is incredible. You can literally watch the cells breathe light. You are watching the clock tick in real time.
You are. And what happened when they ran this temperature cycle on the glowing cells was amazing. The glowing peaks were perfectly inverted.
Meaning what?
Meaning the mouse cells and the human cells entrained, they synchronized their internal clocks to the external temperature to completely opposite phases.
So when the mouse cells were glowing their brightest, the human cells were at their dimmest, and vice versa.
Exactly. The timing was completely flipped.
Okay, let's do a quick recap of where we are so far, because this is foundational. We're about a quarter of the way through our deep dive, so here is the summary. Bullet 1, the master clock in the brain is exactly the same across all mammals. It loves the light.
Right.
Bullet 2, the peripheral cells in the rest of the body have a built-in signal inverter. And bullet 3, they react to the daily rise and fall of body temperature in completely opposite ways depending on whether the animal is nocturnal or diurnal.
Spot on that.
Which leads me to ask you, the listener, a question. Have you ever considered that the skin cells on your arm or the liver cells in your abdomen might be experiencing the time of day completely differently than the brain inside your head?
It's kind of trippy. It forces us to view our own bodies as a collection of somewhat independent time zones.
Yeah, it's like a biological time zone map.
And it naturally pushes us to the next massive question. If the clock genes themselves are identical and they just glow at different times, what exactly is the cellular inverter causing this opposite reaction?
Where's the physical switch?
Right.
Well, if the issue isn't the clock itself, logic dictates it has to be the factory that builds the clock.
And the researchers followed that exact logic. They shifted their focus away from the circadian genes and looked at global protein synthesis.
The actual manufacturing process.
Yeah. They wanted to see how the literal translation machinery of the cell, the factory turning RNA into proteins, reacted to heat. And here's where they found a massive divergence.
Okay, I'm ready. What did they find?
When you heat up a mouse cell, its overall protein manufacturing speeds up. But when you heat up a human cell, astonishingly, its protein manufacturing either slows down or just completely ignores the heat.
Okay, wait. I have to push back on this. This sounds like it breaks the fundamental laws of basic chemistry.
How so?
Well, simple kinetics tells us that heat speeds up all chemical reactions. You boil water, the molecules move faster, they bump into each other more frequently, reactions happen quicker. How can human cells just defy the basic laws of kinetics and decide to slow down when things get warm?
Human cells are not defying physics. They are responding to a different branch of physics altogether. They're responding thermodynamically rather than kinetically.
Thermodynamically. Okay, that sounds like a heavy distinction. Let's lay that out clearly. What is the actual difference inside the cell?
Think of the interior of a cell like a crowded dance floor in a nightclub. The mouse cells are reacting purely to the tempo of the music.
Okay, the kinetic energy.
Exactly. As the room heats up, the music plays faster, the molecules bump into each other more often, and the mouse cells just dance faster to keep up with the tempo. They speed up.
Okay, so the mouse is just riding the kinetic wave. What about the human cells?
The human cells are reacting to how sweaty and physically crowded the room is getting.
Oh, wow.
Yeah. The interior of a cell is packed with giant macromolecules, proteins, RNA, complex structures.
Yeah.
As the physical temperature increases, the behavior of water changes.
The water inside the cell.
Yes. More water molecules become trapped or bound within the hydration layers of those giant macromolecules. This severely reduces the amount of free water swimming around in the cell.
Wait, let me make sure I'm getting this. Biologists have a term for this, right?
Yeah, it's called a drop in water potential.
A drop in water potential. So the physical space inside the cell actually changes. It suddenly feels much more viscous and crowded, even though the total amount of stuff hasn't actually changed.
Exactly. The dance floor suddenly feels packed, shoulder to shoulder. And human cells have a sensory system that detects that exact physical crowding.
A crowding sensor.
Yeah. They use two major biological pathways to monitor this. The first is a group of proteins called WNK kinases.
WNK.
Yes, WNK kinases. They are essentially the master sensors of cellular water balance and osmotic stress. The second pathway is mTOR, which stands for the mechanistic target of rapamycin.
mTOR. I've heard of that in longevity circles.
You definitely have. You can think of mTOR as the foreman of the cellular protein factory. It regulates all protein synthesis.
Okay, let me make sure I'm visualizing this correctly. The human cell gets hot. The water gets trapped by macromolecules. The inside of the cell feels physically jammed.
Yep.
So WNK and mTOR sense this traffic jam, and instead of dancing faster like the mouse cell, they tell the protein factory to hit the brakes.
Exactly.
They intentionally slow down to save energy and stabilize the cell, completely overriding the kinetic urge to speed up.
You have the mechanism perfectly. Mouse cells react to the sheer kinetic energy of heat by speeding up. Human cells, our diurnal cells, possess a thermodynamic shield.
A thermodynamic shield.
Yeah, they react to the physical crowding caused by heat, using the WNK and mTOR pathways to actively buffer themselves and hold their protein synthesis steady.
Okay, time for another quick check-in on our journey. We are deep into the mechanics here.
It's a lot to process.
It is, so let's summarize. Bullet 1, mouse cells react to heat by just going faster kinetically. Bullet 2, human cells are heavily buffered thermodynamically. And bullet 3, we use WNK and mTOR to sense thermodynamic crowding, acting as a shield to stabilize our cellular factories when the fun beats down on us.
That's the perfect recap.
So listener, think about this. If your cells are actively using a thermodynamic shield to buffer against temperature, what happens if we pull a thread? What happens if we chemically or nutritionally break that shield?
And that leads directly to the next phase of the research. Because if mTOR and WNK are the molecular secrets to this diurnal shield, can we manipulate those pathways to change a mammal's fundamental nature?
Can we take a creature of the dark and force it to walk in the light?
Exactly.
So wait, if this thermodynamic shield is just the human cell reacting to being crowded, is there a way to trick it? Can we clear off the dance floor and see what happens?
The researchers actually started with a chemical hack in a petri dish to see exactly that. They took mouse pituitary and lung tissues, which normally speed up when heated, and they applied a specific chemical called INK-128.
INK-128, what does that do?
It is a targeted inhibitor that blocks the mTOR pathway. It essentially gags the foreman of the protein factory.
Oh, wow. And what did the mouse tissues do without their foreman?
The results were immediate. By inhibiting mTOR, the mouse tissues suddenly delayed their circadian phase by 8 to 12 hours.
A 12-hour shift, that's half a day.
Exactly. The timing of their glowing TER2 proteins completely shifted. In that petri dish, the nocturnal mouse tissues started acting exactly like human diurnal tissues. They inverted.
That is wild. Okay, but using a specialized chemical in a petri dish is one thing. Did the paper say if there is a way to change this in a living, breathing animal? Can you hack a whole mouse?
You absolutely can. And they did it using dietary hacks to suppress mTOR naturally.
Dietary hacks?
Yeah, they utilized two different methods on live mice. The first was a work-for-food paradigm.
Meaning they had to exercise for it?
Yes, they forced the mice to run on a wheel for hours just to earn a single pellet of food. This puts the animal in a state of severe caloric restriction and low energy.
That sounds intense. What was the second method?
The second method was a highly specific diet called an isocaloric methionine-restricted diet.
Let's slow down on that second one. Isocaloric just means the total calories stay the same.
Right, they aren't starving them.
But why pull one specific amino acid, methionine, out of their diet? Why does changing one building block alter an animal's perception of time?
It goes back to our factory foreman, mTOR. Methionine is an essential amino acid, but more importantly, it is one of the primary triggers that activates mTOR.
Oh, I see.
Yeah, it is the foreman's favorite building block. When there is plenty of methionine around, mTOR keeps the factory running at maximum capacity. But when you restrict methionine in the diet, The foreman effectively goes to sleep.
The factory slows down.
Exactly.
And when the factory slows down, the physical crowding inside the cell drops. The cell is chemically tricked into feeling uncrowded and cold, which mimics the nighttime environment the mouse prefers, but it's happening during the day.
You nailed it. And the behavioral results of tricking those cells were stunning. By putting the mice on these mTOR-suppressing diets, these strictly nocturnal animals actually shifted their running activity and their core body temperature rhythms straight into the daylight hours.
They became functional daywalkers.
Yes. By nutritionally dampening the mTOR pathway, the researchers successfully flipped the biological switch.
That is mind-blowing. They changed the fundamental behavior of a mammal just by tweaking its protein factory.
It's incredible biology.
But it makes me wonder, is this just a clever lab trick, or did nature actually do this on its own? Did evolution use these exact same pathways when our ancestors stepped out of the shadows after the asteroid hit?
To answer the evolutionary question, the researchers turned to the Zoonomia database.
The Zoonomia database?
Yeah, it's a massive comparative database containing the genomes of 242 different mammalian species. They wanted to see if the genetic code backed up this cellular switch.
Comparing 242 distinct genomes is a massive undertaking. What did the DNA show them?
The DNA showed a textbook example of convergent evolution. They looked at various branches of the mammalian family tree, species that had independently evolved from nocturnal ancestors to become diurnal. And across the board, they found that genes in the mTOR pathway, specifically genes like RAGB, ZNF598, and TSC2, and genes in the WNK pathway like WNK1 and WNK4, evolved significantly faster in diurnal mammals compared to their nocturnal cousins.
So different species all hit upon the exact same genetic upgrades to step out into the sunlight.
Precisely. They all upgraded the factory foreman and the water sensors.
The paper also mentioned something really specific about the actual shape of these proteins. They looked at the WNK1 protein, that master sensor of water crowding, and they found that in diurnal mammals, it has a lower intrinsic disorder.
Yes, that is a key structural detail.
Can you explain what intrinsic disorder means in a protein? What does a floppy protein actually do?
Okay, so proteins are long chains of amino acids that fold into specific 3D shapes. Sometimes, parts of those chains don't fold tightly, they remain loose and flexible, which biologists call intrinsic disorder.
So they're just sort of flopping around like cooked spaghetti.
Exactly. And a floppy protein is highly sensitive to changes in its environment. When a cell gets hot and crowded, a floppy protein is very likely to lose its shape, accidentally clump together with other proteins, and trigger a massive chaotic stress response in the cell.
Basically, it panics when it gets hot.
Yes, it panics.
Oh.
But diurnal mammals evolved a WNK1 protein with lower intrinsic disorder. It is structurally tighter, more rigid.
Oh, that makes sense.
So when a human cell gets hot and crowded in the middle of a summer day, our WNK1 protein holds its shape. It doesn't clump, it doesn't canic, it calmly signals the factory to slow down. It is literally built at an atomic level to be more robust against the heat of the day.
Let's lock this segment in with another summary, because we just covered a ton of ground.
Let's go.
Bullet 1, inhibiting the mTOR pathway, whether through drugs like INK128 or specific diets like restricting methionine, forces nocturnal cells to behave diurnally.
The factory slows down, and the animal becomes a daywalker.
Right. And bullet 2, we know nature did this deliberately because comparative genomics from 242 species prove that diurnal mammals specifically evolved tighter, less panic-prone WNK and mTOR pathways to handle the heat.
Exactly.
It really makes you wonder, doesn't it? Could the specific macronutrients in your diet right now, today, be directly altering how your internal cellular clocks perceive the time of day?
And that question brings us to the ultimate takeaway of this entire field of research. We have to look at the CTE hypothesis, the circadian thermoenergetics hypothesis.
Circadian thermoenergetics.
Yeah, it answers the ultimate why of this entire evolutionary journey.
Right. Why bother evolving this complex thermodynamic shield at all? Why didn't we just stay in the dark?
It comes down to basic survival economics. It is all about energy. Homeothermic, meaning warm-blooded, mammals burn a massive amount of metabolic energy just to maintain their internal body temperature.
That makes sense.
Being awake, active, and hunting during the cold, dark night requires an exorbitant amount of calories just to keep from freezing.
So switching to the daytime, becoming diurnal, was actually a genius evolutionary energy-saving measure.
Absolutely.
If you are active during the day, you use the ambient radiant heat of the sun to help warm your body. You save your own precious metabolic fuel.
The ambient heat does the work for you. And at the microscopic level, that human cellular buffer we've been talking about, are WNK and mTOR pathways resisting thermal shock, keeping protein synthesis steady instead of racing out of control, is the molecular manifestation of that energy conservation.
So the cells don't burn out.
Right. Our cells evolved a way to not waste energy hyperreacting every time the sun beats down on us.
So what does this all mean for you, the listener? It means your biology is a living, breathing artifact of surviving the post-dinosaur world. You are hardwired to save energy.
You really are.
Your cells use complex thermodynamics, something the literal physical crowding of water molecules, to keep your internal clocks ticking steadily, regardless of how hot you get walking around in the afternoon.
The mechanisms of survival from millions of years ago are still dictating how our cells manage energy and time today.
Which brings us to one final, incredibly provocative thought to leave you with. We just discussed how caloric restriction and low methionine diets, diets that lower mTOR activity, can make a nocturnal animal behave like a diurnal one.
Yes.
Restricting those nutrients stabilizes their clocks and tells them it is safe to be in the light. But look at the modern human world we have built for ourselves.
That's the exact opposite.
Exactly. We live in an environment of constant, overwhelming calorie abundance. We eat high-protein diets rich in methionine. We snack constantly from the moment we wake up until midnight. All of these modern habits do one specific thing. They keep our mTOR pathways permanently, chronically spiked.
We are keeping the cellular factory foreman awake and screaming at maximum volume 24 hours a day.
And that is what I want you to mull over today. By keeping our mTOR pathways constantly fully activated through our modern diets, are we secretly confusing our ancient, hard-won diurnal clocks?
It's a scary thought.
By never letting the factory slow down, are we inadvertently overwhelming that thermodynamic shield and telling our cells, at a molecular level, that it's time to go back and scurry in the dark?
The predators from the Cretaceous period might be long gone, but the biological choices our ancestors made to avoid them are still echoing in every single bite of food we take.
Definitely something to think about the next time you find yourself reaching for a midnight snack. Thanks for joining us on this deep dive into your own cellular history. Catch you next time.